So dear colleagues and friends, welcome to UDG. Today we have a great pleasure to welcome and listen to Professor Onur Mutlu. It was very hard to summarize all the success that Professor Onur has achieved, but I hope I have managed to emphasize the most important. Professor Onur is a professor of computer science at the Department of Information Technology and Electrical Engineering in Zurich. He is also a faculty member of Carnegie Mellon University. He obtained his PhD and master degree at the University of Texas in Austin, and bachelor degrees in computer engineering and psychology from the University of Michigan. His current broader research interests are in computer architecture systems, hardware security, and bioinformatics. A variety of techniques he, along with his group and collaborators, has invented over the years have influenced industry and have been employed in commercial microprocessors and memory storage systems. He started a computer architecture group at Microsoft Research and held various products and research positions at Intel Corporation, Advanced Micro Devices, VMware, and Google. He received the Huawei Olympus Mons Award, Computer Society, Edward McCliskey Technical Achievement Award, Maurice Wilkes Award, Computer Society Young Computer Architect Award, Early Career Faculty Award, National Science Foundation Career Award, Carnegie Mellon University Research Award, Faculty Partnerships Awards from various companies, and healthy numbers of best paper and top big paper recognitions, etc. This is just a part of his achievements. He's an elected member of the Academy of Europe for outstanding achievements as a researcher. He's one of the top cited scientists in the world. I truly believe that all of the above stated is a strong motivation to carefully listen to today's lecture, and it will be the inspiration for us to start a fruitful discussion afterwards. Dear Professor Onur, thank you once again for being with us, and the floor is yours.
Thank you very much for the time. Thank you. Thanks for coming, and thank you for the nice introduction. I'm happy to be here. It's my second time in Montenegro, and first time at a university in Montenegro, I should say. How many people here are students? Undergraduate students? Some graduate students? Okay, some. How many people are professors? Okay, there are some professors. Okay, we have a good mixture of people. And you all belong to UDG, the university. Anyone from outside? Well, I know Velko is from outside, but okay, some people are from outside. Where? From industry? Okay, other universities. Fine. Okay, sounds good. Okay, everybody is welcome, of course. Before I start, how much time do we have for the talk? 1 hour? 1 hour. 1 hour plus questions after that, or? Okay, the reason I ask is I have a lot of slides, as Velko knows. I can talk about this topic for days, but I don't want to talk about it for days. So please stop me when you think I'm going over. I will cover the most important information early, so you can go to sleep when it becomes boring for you. I know that sometimes students don't listen to presentation unless there's an exam following the presentation. I don't know if there's such an exam here, but I'm not going to give you an exam. So let's get started. So I'm going to talk about something that's very exciting for me and for my research group and for many people in the world, which is accelerating genome analysis. This is a journey that I have been taking for, let's say, 17 years. And right now, I think we're experiencing a lot of really interesting things in genome analysis. And hopefully, I will share some of them with you. Maybe you will be inspired to do something big in this area. Who knows? That's we can only hope. So let me give you what I'm going to talk about. So we're going to talk about bioinformatics. Bioinformatics is information technology and biology put together, processing of biological data, essentially. And this is an important problem, especially if you need to design systems to do this processing. And it clearly has large scientific, medical, societal, and personal implications. I'm going to give you some of these to begin with. And this talk is going to focus on a key step in bioinformatics, which is accelerating genome sequence analysis. So it's not, bioinformatics is a much bigger field, actually. We're going to focus on genome sequence analysis. And we're going to focus on even a smaller portion of genome sequence analysis. So you will see that it's an important portion, but there's a lot more out there in bioinformatics. So in particular, we're going to talk a lot about read mapping. So the key takeaway is many bottlenecks exist in accessing and manipulating large amounts of genomic data during analysis. And there are many recent ideas that we have developed to accelerate read mapping and beyond. Actually, this talk covers a little bit beyond as well. And I'll give you my personal journey since September 2006, if you will. That's when I started actually at Microsoft Research. After I finished my PhD, I went to Microsoft Research to form the computer architecture group. And I was very interested in genomics over there. And there was a local university over there, University of Washington, and they had a nice genome sciences department. And I had some friends out there, and we together started this research direction at the time. So what did we discuss at the time? While we were hiking in the mountains over there, and Mount Washington is a nice mountain, Mount Rainier. If you go there, definitely visit it. Basically, we were dreaming about this. And our dream was basically having a small device, embedded device, that can perform comprehensive genome analysis within a minute or less than a minute. At that time, genome analysis was taking like sometimes months, depending on the analysis you do, of course. So we're going to talk about that. So this could be questions like, which of these DNAs does this DNA segment match with? This is actually interesting for understanding a genome like COVID RNA, for example. Right? It doesn't have to be DNA, also it could be RNA. Right? It could be proteins, actually. What is the likely genetic disposition of this patient to this particular drug? Clearly, this has important implications for that patient, at least. Right? What disease or condition might this particular DNA or RNA piece associated with? And there are many, many other questions. I'm going to give you some more questions also. These are more medically oriented questions, but they're also scientifically oriented questions for understanding evolution, etc. So we wanted to do this. Clearly, genome analysis actually has a lot of other things, like you can predict the presence of microbes in a sample. You can survey disease outbreaks, like COVID-19 could have been predicted potentially. You know what personalized medicine methods. You can understand evolution, genetics, etc. We're going to look at these in a little bit more detail soon. But the takeaway is that there's a lot to do over here, and we have sequencing machines that look like this. It's a nanopore sequencing device, both relatively cheap, $1,000 or so, compared to other machines that cost $1 million, let's say. You can actually do this. Say it. Oh, you have it. Okay, that's great. You have it. So you can actually do the studies. How many people are working on this here, like nanopore sequencing devices? Okay. Okay, great. So there are people who are working on it. So you can probably sleep through the background slides. But I'm going to give you some background on this. If you're not working on it, that's not a problem. So basically, for those who haven't know that this actually powerful, even high school students today use these devices to analyze fish genomes, for example. There are stories about that people report firsthand. So I'm gonna. So if you don't want to listen to this, there are a bunch of overview readings that we have actually put out. We keep writing these overview articles because this is a field that's not easy to get in. Like when I was first starting, there's a lot of terminology in bioinformatics in general. And if you have a computer science background or electrical engineering background, not easy to get in. So we wanted to make it easy for people to get in. So if you're interested, you can look at these articles. I'm going to talk about some of these. This is the article that we wrote in this invited issue of this biotechnology journal, which is interesting. And this is a very short article that's coming up at DAC in July. So if you want to read something really short, this is the thing to read fast. Okay, so let me also give you the bigger picture today. So that nanopore machine was small, right? And this is $1 million machine. This is a big machine that actually you put DNA into it and it can sequence it fast and high throughput. But this is a very special purpose machine that can generate data really well. It's very fast. Unfortunately, it has no analysis capability, meaning those questions that we ask, it cannot answer them. You have to move the data significantly to some other machine, and usually to a cloud data center, and use general purpose machines to analyze the data. So you can see the mismatch in this picture. This is very specialized, a lot of data moments, and then you try to process the data with a general purpose machine, which is fundamentally slow because it's general purpose. It's not special. So we have this mismatch today in genome analysis. We have extremely good sequencing machines. We have, let me put it bluntly, terrible processing machines, if you will, compared. So that's what we're trying to bridge. We're trying to get this processing machine to be almost as good or even better than these machines. and minimize data movements that we're causing, because data movement fundamentally is energy inefficient and also very costly in terms of performance. I should also say that this is not necessarily just in genomics. Genomics is very different because these machines are powerful. But if you think about video cameras, you can actually replace video cameras with this machine also. Once you have video cameras, we do the same thing. Video processing is very specialized. Well, video capture is very specialized, but unfortunately, processing is not so special. And also, you can add other data generators here, but I don't have time for it. So basically, that's why we're looking at this problem from an algorithm device co-design perspective. We want to design algorithms that are extremely efficient, that the machines are good. And also, we want to design a hardware that matches those algorithms such that it's very efficient. Because in the end, these machines are hardware machines. They're not software machines. They're actually generating signals that you're analyzing the signals. Okay, we're going to look into more of it. So that's a high-level picture. I gave you the high-level messages. Now let's jump into the agenda. So how many people have any background on genomics here? Biology. You heard of what a genome is, DNA is, maybe. Okay. Yeah, all of you have some DNAs, I can guarantee you that. But yeah, so it's good to think about. So basically, we have genomes and we all have DNAs. Viruses have RNAs. Basically, all have also RNA. But basically, it consists of a bunch of bases. My intent is not to give you a biology background here, but these consist of A, C, G, T bases. You can learn more about these. These are real. DNA actually determines your propensity or some traits and what you look like, for example, in the end, because there's a process that happens. DNA gets replicated, gets transcribed into RNA, and proteins are made out of RNA through translation. Essentially, this is the cycle of life, if you will, and there's a lot of information you can think about. DNA, you can think of it as a cookbook, and in the end, you make some pie out of that cookbook. Right? And this is real. And this is actually a very famous DNA from Henrietta Lacks, whose DNA was taken from her without her consent. So there are a lot of privacy issues surrounding the DNA analysis also, which I'm not going to talk about, but you should think about that. This led to a lot of understanding of the DNA. Something that was taken from someone without her consent actually led to a lot of understanding. So, okay, DNA looks like this if you turn it into text. Basically, it's a bunch of text characters, ACGTs, right? These are called bases or base pairs because they come in a paired way, which we're not going to talk about. And essentially, the idea is, if you look at a human text, human DNA, it's 3.2 billion base pairs. And some of them are important. Basically, this name over here, they actually determine that you have high blood pressure, right? Or this sequence over here may determine that you're more vulnerable to Down syndrome or something like that. Some of these are well understood, as I will show you. So basically, how large is this text? Human is not the largest, as you can see, it's 3.2 billion base pairs, but there are some things that are much smaller, viruses, and there are some things that are much larger, 149 billion base pairs. So we don't quite understand exactly the length and other relationships, but we can guess what's happening by analyzing the genome. So we're still building a lot of understanding. So the goal in DNA sequencing is to figure out that text essentially, find the complete sequence of ACGPs in an organism's DNA or RNA. The challenge is, unfortunately, we don't have a perfect machine today. Our machines are powerful, but there's no machine that takes DNA as long inputs and gives the complete sequence as outputs. Machines today are choppers. They chop DNA into pieces, usually random pieces, and they give you small pieces. They identify small pieces, but they don't tell you how they fit together. So immediately, the problem is, what does the entire piece look like? So if you have 3.2 billion base pairs, some of these machines are going to chop them into 300 base pairs each. So you have lots of things, and you'll try to you have a problem of stitching in the end. So genome sequencing essentially tries to do this. Basically, you have a large DNA molecule. It gets chopped into these small DNA fragments, and then I think it's slow, so maybe okay. And these small DNA fragments are called beads. And the idea in genome, oh, this is slow, so maybe I'll go over here. This may be a better place. Yeah, basically, current sequencing machines are all like that. They provide fragments of this original DNA sequence. So I call this, I liken this to a puzzle. Essentially, you have this genome. This is what you would like to obtain in the end. But when it's chopped into pieces, you have this puzzle that's all around. And you want to actually reconstruct this puzzle. But this is not a perfect analogy because
when the machine chops the DNA into pieces, it actually inserts a lot of errors. There are errors that are happening. So it's not a perfect matching process. And we will talk about that in a little bit. This is a better analogy, perhaps. I liken this to untangling yarn balls. So you have these yarn balls your cat may be playing with. And the idea is actually the chopping machine goes through them and chops them into pieces. And you have a lot of different fragments of DNA that you're trying to figure out which yarn ball it belongs to and exactly how it fits. You're trying to reconstruct that yarn ball, essentially. So these are the chopping machines that we have today. Some of them are old, some of them are new, some of them are extremely capable, like this one over here. Some of them are a collection of these small nanopore machines. Basically, you can put many of these together and it becomes a data center like genome sequencing machine. So basically, these are machines with different properties and they produce data with different properties. These are more of them, as you can see. Essentially, these machines have enabled the genomic era. So initially, human genome was sequenced and it was very painful. It was expensive. I'm going to talk about that briefly. But because of the introduction of these high throughput sequencing technologies, which were the machines that we have seen earlier, the large ones, the cost of genome sequencing has been reducing significantly. And the cost has been reducing exponentially, as you can see. This is the introduction of high throughput sequencing. This is the introduction of nanopore sequencing. The cost has been, there have been inflection points. Whereas the cost of a transistor has been reducing less, much less. This means that if you actually try to put transistors to work and throw transistors at the problem, you're not going to match the cost at all, because sequencing machines are becoming much more powerful than our computers are becoming powerful. Okay, so this is the same picture. That picture ended in 2015, but basically the trend is continuing. You can see that we have a problem, let's say. So this is what I mentioned earlier, right? You have this special purpose machine that's scaling very nicely. This general purpose machine, just by looking at this picture, is not scaling nicely because it's dictated by Moore's law. So the question is, what are we going to do about this? So this is a data generator. We're going to talk about the nanopore machine which we have. Essentially, revolve it. Oh, this is okay. This is one of the machines. I'm going to stay here because this is going to be my backup plan. When I go farther, it's not going to. It's not working very well. So you can see that the data mode problem is just over here also. This thing is trying to communicate over here. The distance actually matters, and it's a lot easier to process things locally over here. So that's the gist of my talk, basically. Let's process things locally in a specialized way, as opposed to moving data around. So okay, this machine is a great data generator, but you have to move the data around a lot to actually process the data. So we're going to talk a lot about that. But this machine actually generates data that is different from other machines. So it generates long reads. Long reads are 50,000 kilobits or sometimes millions. It's increasing, but the accuracy is lower. There are some other machines that are extremely accurate. Accurate meaning what you get out of the machine is actually what the reference genome looks like. ACPGs are correct. Sometimes you get incorrect measurements because of the way the machine operates. There's variation and errors in the machine also. So some machines are extremely accurate, but they generate short reads. Some machines are not so accurate, but they generate long reads. And ideally, you would like to generate as long read as possible with perfect accuracy, right? Because that's what you really want in the end, the full genome. But unfortunately, we have this trade-off in these machines today. Okay, so basically, that's the gist of the genome analysis. You have these sequencing machines. The first step is to figure out what the genome looks like through a technique called read mapping, which we're going to talk about. And then after read mapping, you can actually decide which part of this genome is vulnerable, for example, to a particular disease. You can ask a lot of questions that we're going to talk about. And then you can enable medical advancements, personalized medicine, and scientific discovery. So that's our goal, basically, accelerate the entire process. And I'm going to talk about read mapping, but if you're interested in read mapping, people have been developing read mappers for, let's say, 40 years or so. And this is our recent survey that ends in 2020 because the article was published in 2021. This is a fascinating topic. We analyzed a lot of these read mappers. Don't let this 111 pages scare you. The shorter version of this is actually much shorter. So if you're interested in read mapping, this is a survey, this is a state-of-the-art survey that we wrote with many people who have been working on read mapping for a long time. So before I go into more detail, let me talk about why do we care. I'm going to keep this relatively quick. I have a lot of slides, but I'll try to go through this short. But basically, there are many questions that can be answered if you actually make this process fast and accurate. If I give you a bunch of these sequences, tell me where they're the same, where they're different. This enables me to distinguish the evolution of species, for example, or the familiar relationships between different things. These are some bacteria, for example, and this is a map of how they differ and how they are similar in terms of their genomes. And this is a nice picture that I like using because this, despite all of our differences, this shows that we're very, very similar to each other in the end. However, dissimilar we may look like, but we're also quite similar to a banana. So sometimes you may want to interact with a banana compared to humans, but we should be interacting better as humans also. So, but this kind of questions you can answer basically looking at genetic race. I think there's another reason why I put the slide actually, which is to show you that a lot of the genomes are similar. What you really are focusing on are the dissimilar parts, and that makes this a difficult problem. I don't care about the similar parts in general. I care about the dissimilar parts. I care about what makes this person different from this person. In many different ways, of course. Genome is not the only thing that I should look at, but genome gives me indications of different things, especially related to health and medicine. Right? So, okay, this is an example of blood pressure, for example. This is something we know, and you can read the papers over here if you're interested. This is called a single nucleotide polymorphism. Essentially, a single base may change, or two bases in this particular case, and the change in these two bases in a particular genome in humans lead to significant differences in blood pressure propensity, as you can see. So this is not the only thing, but there's a correlation that you have. If you have this particular single nucleotide polymorphism in that particular position in your genome, your blood pressure may be high. Okay, so you can see that this could be useful, right? There are other things, for example, people do these genome-wide association studies based on sequenced genomes, and they have these experimental data. They compare people without heart disease versus people with heart disease, for example. And these different single nucleotide polymorphism differences in particular place in the genome are correlated with heart disease, for example. Okay, then based on this, you can develop personalized medicine, for example, hepatitis C. There are a lot of interesting things. If you're interested, you can look at the links over here. I'm happy to make these slides available also. And there are also, these are single variations, but also there are larger variations like 593 kilobits, which is an interesting size. But these also have been shown to be correlated with different types of disorders or propensities to obesity, for example, etc. Okay, so there are interesting studies you can do. People report these things in medical journals sometimes. This is an interesting one where they show that if you can actually do these studies for critically ill patients, you can reduce the treatment costs significantly by understanding their genomes. But today, it's expensive, as you can see, right? In some cases, it takes 5 days to actually get the data that you want when you sequence the genome. If you actually want it in 2 days, it becomes extremely costly. Now, the problem is that may not be enough to save the input, right? So 2 days, in these cases, in some cases, even 5 minutes is too much. So that's the goal of what we're trying to do. Basically, we want to reduce those 2 days, which is the aggressive one today, to 1 minute or even less. Can we do it in 1 minute so that we can actually react very fast in cases where we need to react very fast, and we can be efficient in other cases. Okay, so if you're interested, there is I have recommended reading. It's a nice review article that talks about these structural variations in genomes and their different indications. Okay, there are other interesting studies that you can do. So if you, for example, somehow see a bunch of short sequences, I'm going to give you the context of it. Can you identify who does it belong to? Can you identify genomically unknown organisms? This is usually called metagenomics, and people that do these studies called microbiome profiling. So you go through this place and you take samples, crowded city, and take samples and try to figure out what's going on. This could be useful for understanding potential outbreaks that may be coming up, for example, or understanding some variations that happen in bacteria and viruses. And people like to do this. This is a study that was done in New York City, for example, where they actually tried to understand what kind of organisms are lurking around. And a lot of them are unknown organisms, as you can see over here. They don't know what this is, partially because we haven't done enough sequencing, let's say. Okay, and people are trying to do this in a global scale also. And this is one article that talks about that, which is very interesting, but we don't have time to go through this. You can see the amount of data that we need to generate if you actually do this in a global scale. So this is one like Twitter post where people are talking about how many microbiomes. This is called microbiome analysis. So in 2020, we're already at exa, let's say, number of exabytes, let's say. But in 10 years, we're going to be at yottabytes, and I cannot read that number over there, as you can see. I think exa is to the 60, zeta is to the 70, yotta is to the 80. So genomic data is actually growing much faster than any other data that we know of today, if assuming you're going to do these studies, of course. So another question, a lot of you have lived through the COVID pandemic, and these nanopore sequences were very useful in understanding the COVID pandemic. This is actually an article from January 2020, and people were shipping these nanopore sequences to China to really sequence the COVID genome, and they've been very instrumental in understanding. But this is not the first pandemic. There are other, people have actually written a lot of papers about understanding the COVID RNA. But people have used genome sequencers in the past also, like Ebola in the past. But you can see that the setup that they have in Africa, for example, in this particular case, is pretty not so great in the sense that you have these sequencers and then they're attached to these laptops and you're trying to analyze data. And it takes a long time. In some cases, it takes weeks. And if you really want to get something done quickly, it's not happening quickly. We want to make this happen quickly. So I can keep giving you more and more examples, but otherwise we'll be having only examples in this talk. So let's move on, I would say. Hopefully it's clear that the problem is important. Now let me jump into one problem that I'm going to focus more on next, which is really the read mapping problem. Essential sequencing machines are very good. We want to reconstruct the genome after the sequencing machine chops it. How do we do the read mapping? And it turns out we already have a bottleneck immediately in terms of computational power. As I said, this is fast and the computational machines are slow. So we have a huge bottleneck in terms of the throughput of the machine. Okay, so this is the read mapping bottleneck, and you can actually read papers about it. So the machine is very capable, sequencing machine, but the analysis is not as capable. So that's the picture that I showed you earlier. I will not go through this again. So we're trying to eliminate these bottlenecks in the genome analysis pipeline. So one problem is you need to reconstruct the entire genome from many sequence reads or fragments. So let me more formally define what's read mapping. Essentially, we want to map many short DNA fragments to a known reference genome with some differences alone. We want to allow some differences because there are errors in the machine, but also we want to find the variations between humans because there's a reference genome that's represented today. It belongs to some human, but naturally I'm going to be different. My genome is going to be different from that. Your genome is going to be different, and all of us will be different in some cases. And we would like to identify those differences as well as similarities. So basically, logically, DNA looks like this, physically looks like this, actually. We chop it into pieces using these machines called reads, and then we try to map it to a reference genome. So usually constructing a genome that you sequence is done this way by trying to map these small pieces to a large reference genome. Now, how do you construct a reference genome is another question. It could be done partially through these techniques, but there are also other techniques. I'm not going to tackle that right now. That's an even harder problem. Constructing the human reference genome, for example, took 15 years or so with a lot of investments, let's say. Right now, we're much better at it. Once we construct a genome, it's relatively easy to construct some unknown organism's genome, but it takes time. Okay, so in the end, hopefully, you will find the matching parts and find the non-matching parts. So mapping short reads to a reference genome is challenging because you may have billions of 50 to 300 base pair reads. That's a lot. Okay, now this problem becomes even worse when you do metagenomic analysis. Basically, you have some unknown genetic material that you recover from environmental samples, and you try to map these to many different known reference genomes to identify what am I looking at. And the hope is that you're trying to map these reads into a reference database in the end. And some of them map to multiple different genomes, right? So there's a discussion, for example, right now, which is a really weird discussion scientifically, in my opinion, whether the COVID-19 pandemic was caused by raccoon dog in the Wuhan, whatever, the market, right? And people are using a very weak evidence of one particular piece matching a raccoon dog DNA to, let's say, further the theory. It's very weak evidence, in my opinion, but basically, they're doing metagenomic analysis in this case. They're taking samples from this market and they're trying to match it to a raccoon dog. The question is, you could try to match it to anything, and there will be some matches. The question is, how confident are you in those matches, right? Okay, anyway, that was a sideline, but you can see that there are really real scientific as well as political implications because it's a very heavily politicized concept, unfortunately. Yeah, okay, basically, this is the metagenomic analysis. So let's go into a little bit more detail. What does it look like? What does the read mapping look like if you look at the text file? So today you can produce text files. I'm going to talk about emerging technologies going into the future. But this is a text file that you get from a machine. These are some standardized formats. These are sequence reads. So basically, you have some fragments over here, and you have some fragments in the sequence reads that may match those locations. What you're looking for over here is to find this part in the reference genome that kind of looks like this, right? I just colored them, color coded them. So you don't want this part, you don't want that part, you want really this part, because this is the part that's matching it. And you can immediately see that it's not an easy problem, right? So you can do base by base comparison. You have a read and you want to find the portion of reference, compare it. Usually what's done is, this is called read alignment or read verification. You want to find some edit distance. So essentially this is text matching or approximate string comparison. Edit distance is defined as the minimum number of edits, changes essentially, insertions, deletions, or substitutions that are needed to make the read exactly match with the referencing. So you can take a read, go through all parts of the genome and try to figure out the edit distance with all parts of the reference genome. This is an extremely expensive process. It's not done today this way. Today we actually build indices, etc. But fundamentally, in the end, you have to do this. What I will try to convey is that you want to minimize the number of cases where you do this as much as possible. You don't want to do this. You want to actually eliminate most of this and do this only when you really need to do this. Okay, let's take a look at an example. So text, there's Netherlands, there's Switzerland, and this is the edit distance between Netherlands and Switzerland. Essentially, there are two mismatches over here in the beginning. There's one insertion in Switzerland. There's a match over here, which is the yellow part. There's a mismatch over here, and these are matches. And then there's a deletion in Switzerland that doesn't exist in Netherlands. So you can see that there are 1, 2, 3, 4, 5. The edit distance between Netherlands and Switzerland is 5. So this becomes interesting because clearly you need to have a boundary. Right? What is the edit distance? What is the allowable edit distance that you're interested in examining? If you want only 5% to differ between two different texts, that is interesting. If you want 200, like 100% to differ, clearly every text will match. So there is a threshold that you need to look at, and these have medical implications, which I'm not going to go into. But ideally, we would like to be able to say, I want to compare, find the reads that match portions of the reference genome with some edit distance allowed. And I would like to be able to configure that edit distance, 10%, 20%, 5%, 1%, exact match, 0% difference allowed. So I should be able to say that, and I should be able to get those done efficiently and fast. Okay, so the challenge is you need to find the many mappings of each read. So essentially, a short read may map to many locations. How can we find all mappings efficiently is interesting. How do we tolerate small variance of errors in each read? Essentially, how can we efficiently map each read with up to E errors present? Essentially, that's the edit distance, if you will. And you need to map each read very fast at the same time. How can we design a much higher performance read map? So these are the questions that we're tackling, essentially. So why is this slow? If you actually want to compare, find the edit distance, so this is an example showing these two different sequences. Usually it's presented like a matrix over here. Essentially, you need to traverse some matrix. How many people know about dynamic programming here? Okay, you should probably learn about dynamic programming at some point. But dynamic programming essentially enables you to form this matrix and figure out the edit distance by essentially looking at individual pieces over here. So for example, you traverse this matrix over here, and eventually you say that the edit distance between these two strings over here is 3, because you want to find the minimum one along the diagram. Okay, so this is a very expensive computation because there are a lot of data dependencies as you traverse these things, and the entire matrix needs to be computed, even though strings may be dissimilar. So there's a lot of advancement in terms of algorithms to make this faster. In fact, there were some recent algorithms like the weight font algorithm, which is very interesting. So you can actually accelerate this with software methods, but software methods have limited benefits. In the end, we want to accelerate it with hardware as well. So if you do this dynamic programming with Netherlands and Switzerland, I'm not going to do this. You'll have these slides, so you can actually do this on your own. Essentially, you need to look at every single entry, and you need to fill this matrix. So a matrix fill-in takes mn time. M is the length of this string, n is the length of this string. And then you need to backtrace actually to find out what is the shortest or smallest distance. Okay, so we discussed it. Basically, you have a quadratic time dynamic programming algorithm because you need to enumerate all possible prefixes. You can also think of it this way, basically. You're trying to compare Netherlands to all of these possible incarnations of these prefixes. Okay, I've already said these, so I'm going to go through these quickly. Okay, so if you're interested, there's also actually a lot of theory behind this, and people have shown that edit distance cannot be computed in a subquadratic time. There's a beautiful paper that talks about that. And read mapping in the end talks about edit distance computation. And that's why people have been developing so many techniques over the course of 40 years to make this fast. And still, this is taking a lot of the time of this of a read mapper. Basically, most of the time of a read mapper is today taken by sequence alignments. We'd like to get rid of that as much as possible, as I said. So that's, now let's go into accelerating read mapping. Basically, we would like to accelerate this process. Hopefully, I've convinced you that there's the expensive computation that you need to do over here. We'll talk about memory bottlenecks also. So this computation you also need to do if you want to actually compare texts. So I should also say that this is not just for genomics, it's also more broader than genomics. But if you look at genomics, essentially you have a lot of things to accelerate. So I'm going to talk about some of these acceleration methods, but as I said, if you're interested, you can read more papers. Or better, you can actually look at our genomics course, which is all completely online. You can attend it online also. And there are also workshops that are being organized at major conferences like VCOM, which is a major bioinformatics conference. And you can also watch this if you're interested. So let me jump into, I can talk about a lot of algorithmic methods, but I don't really have time today to talk about those. We've been actually working on both software methods as well as hardware methods. More importantly, co-design methods today. I'm going to jump into a little bit of hardware methods. But before I jump into hardware methods, I also need to talk a little bit about software to give you the insight of what we are trying to accelerate. So this is one of the first works that we have published on accelerating genome sequence alignment using FPGAs. So the overarching key idea is this. Basically, you don't want to do this costly edit distance computation. So filter past before you align. Minimize costly approximate string comparison edit distance computation. In other words, you have some sequence reads. Some of them are too dissimilar from the reference because their edit distance is larger than the allowable threshold, E, that I mentioned earlier. Figure those up quickly and don't do costly computation on them because these are not interesting. But those that are similar to the reference, focus your effort on them. Essentially, do the edit distance computation. These are the interesting parts. But they're similar, but they're not completely similar also, right? If it's completely similar, it's easy to find that out. If it's dissimilar slightly, those are the hard ones to figure out. So our goal is to actually, in this work, is to find quickly what are too similar to the reference, too dissimilar. So we actually had some pure software approaches, which were published earlier. I'm not going to go through this, which is interesting. It accelerates things fast. And then we actually came up with this shifted timing distance metric, which is an alternative to edit distance, especially if you want to find out quickly these things. You don't want to compute edit distance perfectly, but you want to quickly find out the things that are different. So let me give you the insight. Insight is nice. It's from a city that I like. Basically, you have these two strings. One is, let's say, correct. The other one, well, both of them are correct in this case, right? This is good. So basically, if you compute the Hamming distance between these two, you get 8 matches and 0 mismatches. And basically, it says these are exact matches. That's good. Now let's delete A over here. This is one deletion. Edit distance should be 1. Hamming distance is easy to compute. You do XOR, right? And then an AND in the end. So right now, if you actually look at the Hamming distance between these two strings, assuming you pad one of them, you get 3 matches, 5 mismatches. This doesn't sound good, right? Even though these strings are actually quite similar to each other, just as a human. So what we do is basically shifting. To cancel the effect of this deletion, you need to shift the string to the right, 1 character. Now let's take a look at that. Basically, shifting a string helps similarity search. These are the 3 matches, 5 mismatches without the shifted version. Now, to cancel the effect of this deletion, we shift the string to the right. It becomes like this. Now you get 4 matches over here also. So basically, the process of shifted Hamming distance is that essentially you create shifted versions of one of the either reference or read string, and then do Hamming distances on both of them. And then Hamming distance is essentially simple XORs between the characters, as we've discussed. And then you do essentially this is what you get after you do the Hamming distances, or it's a bit slow. And then you AND these Hamming distance masks. And once you AND them, you get only 1, 1, which is basically the mismatch over there. So basically, you count the ones after that, and then you figure out that there's only 1 difference here. That's the idea over here. It's a very simple technique. It's bitwise operations, and you can accelerate this beautifully using something like an FPGA reconfigurable architectures, essentially. So that's the idea of the metric. That was introduced by that paper, and it's actually, if you actually look at it from the matrix perspective that I showed you, there are no data dependencies over here. It's beautiful, and these are the different Hamming masks as we introduced them. So as I said, we did this in software using SIMD accelerators and a general purpose machine, you get acceleration. But with an FPGA, you unleash the acceleration significantly. And that's the idea of gatekeeper. I'm not going to go through this in detail. I'll give you the key results. Essentially, this is one of the fastest filters at the time. You can see the numbers over there, and you get 10x, an order of magnitude speed up in read mapping if you actually design a read mapper using this FPGA elements, which is quite significant. And it's also freely available online. It's a bit old right now. We have better read mappers that I'm going to, better accelerators that I'm going to talk about, but you can also test it. So essentially, the conclusion of this early work is that you can get significant speed ups, also energy efficiency improvements, and you can also integrate this with the sequencer. So I'm going to, at the end, if you have time, I'm going to talk about what's happening in industry today. Industry is actually doing something like this today. Illumina has a Dragon platform. Essentially, they do FPGA acceleration. So basically, our vision at the time was we can enable real-time filtering while sequencing itself. And this could be a way of enabling on-device genome analysis, as I kind of alluded to earlier. Okay, if you want to know more about technical details, but you can read the paper that was published in Bioinformatics. So this is an example of algorithm architecture co-design, I would say. Essentially, we designed the algorithm such that it could be nicely accelerated with a specialized architecture. So what else can you do becomes interesting. This is something that we did in between. I'm going to skip this. It's also interesting, but it's kind of superseded right now. I'm going to give you what superseded it. If you're interested, you can look at this one, but we don't have time. So I'm going to give you the state-of-the-art filtering algorithm. As far as we know, this is the state-of-the-art. It's called Sneaky Snake. You'll hopefully see the reason. So if you look at two different things that you're comparing, and these are from some seminal papers from 1985, essentially, this is one genome and this is another genome, let's say. Correct alignment is really a sequence of non-overlapping long matches. You really want to find these long matches. Our key idea in this work is to get inspiration from some other field, hardware in this case, and we reduced the approximate string matching problem to the single net routing problem in the VLSI chip layer. So you can ask why, how is this relevant? I'm going to hopefully give you the insight without going into a lot of detail. So essentially in VLSI chip layout, you have some input pads into a chip, and then you have some output pads. And then you go from input to output, and you try to find the longest possible wires without getting into obstacles. Basically, you want to route directly as much as possible, but there could be some obstacles along the way because you may have an adder over there, right? That has some metal implications. So the idea is to minimize the number of hops that you need to reroute from input to output. So I will go into a little bit more insight, but essentially, if you reduce this problem to this VLSI net layout problem, you get 4 orders of magnitude more accurate filters. And also, you can greatly accelerate state-of-the-art CPU-based sequence aligners. So you can see the numbers that they're between 1 to 2 orders of magnitude, getting closer to 3 orders of magnitude in some cases. Again, we're using hardware acceleration compared to CPUs. That's why you're getting this. So let me give you the key insight. So the key insight actually goes back to what I just told you earlier. Basically, you have these different diagonals. I showed you in matrix form, but I show you in this form right now. Essentially, you have this string that you're trying to match over here, and you create different diagonals over here, and we basically build a neighborhood map based on these different diagonals. And the edit distance, maximum allowable edit distance is 3 in this particular case. So let's take a look at it. You can actually compute all of these to see what is matching and what is mismatching. Essentially, basically what we're comparing is these 12 over here to these 12 over here, and these are shifted distances, shifted timing distances that I showed you earlier, done in a way that looks like this, essentially rows and columns. So main diagonal, for example, compares the strings exactly. The first upper diagonal compares the second string shifted to the right once. The first lower diagonal compares to this string shifted to the left once, and you can actually generalize it. So if you do that, you can compute all of those matches, and the black ones are saying mismatch, white ones are saying match. Now, this looks interesting. This looks like an entrance to the chip and then exit from the chip. And then your goal here is to maximize minimize the black ones that you miss. And that's the VLSI net routing problem. If you actually look at this, you have the snake that is trying to maximize how many white boxes it traverses before it reaches a black box. Here it goes 4. So that's 1 edit distance reduced. And then you look at the next ones, you go another 4 over here, and then 1 more edit distance reduced. And then you look at the next one, you go 1, and then 1 more edit distance reduced at the end. So in this case, with an allowable edit distance of 3, you can do this. Now, I'm not going to go into the details, but you can actually build all of this on the fly without requiring a lot of memory. So that's the interesting part, and you can put it in an FPGA, and that leads to the huge speed-ups that we have. Does that make sense? I know I gave you a very high level, but it's essentially a lot of algorithms that you need to deal with in the end. If you're really interested, you go and work with the paper, and people have actually built on this, and they've improved some parts of it recently. Okay, no, sorry. I pressed the wrong button, I think. Okay. Okay, so I could keep talking about a lot of these algorithms. I'll mention that more recently, we've been working with Intel, for example, to actually realize this in an application-specific integrated circuit. This is work done by my last PhD student at CANU UDG. She actually looked at approximate string machine acceleration. Let me give you the key ideas. Here, again, our goal was to find some algorithm that's actually really good for acceleration. The prior algorithms are interesting, but there were some algorithms that we found for text search that were developed in 1990s. It's called the White-type algorithm. And we said that, okay, this algorithm looks very interesting also for similarity search and genomic analysis, also for text, of course, because it's very bitwise, simple bitwise operations. And you can actually perfectly compute edit distance using this in some cases, if you put enough hardware. So it's very highly parallel. We basically modified this algorithm. We made it highly parallel. And essentially, we enabled it to perform traceback, which is needed for edit distance computation in the end. And we co-designed it with hardware accelerators. So this is what the accelerator in the end looks like. I'm not going to go through the algorithm because it's too complex to cover it right now. But basically, we have a distance calculation accelerator that uses bit vectors. And then it communicates with a traceback accelerator. And in the end, you get the final edit distance nicely. If you know about systolic arrays, this is essentially systolic array-based acceleration. Which is used in machine learning today also. So you can actually use some of the machine learning accelerators for this purpose, in my opinion. But I think approximate string matching is a little bit more difficult because of the traceback that we will have in the end. Okay, I don't have time to go into detail, but traceback acceleration is very interesting. You can take a look at it if you're interested. In the end, it requires some logic. Let me give you the key results. So compared to a Xeon CPU core, we collaborated with Intel, and they gave us very interesting numbers. We can actually do this with only 1% additional area and power on top of a Xeon CPU core. So it has low area and power overheads. And it can actually accelerate many different parts of genome analysis pipeline. So pre-alignment filtering was what we talked about earlier, and also aligned edit distance computation, and also some other parts that I'm not going to talk about. So essentially, we looked at these different use cases. We also talk about other use cases in the paper, which we don't evaluate. But in the end, compared to the best prior software and hardware accelerators, some of you may be using Minimap2, for example, you can see significant speed up, 2 orders of magnitude speed ups, and between 1 to 2 orders of magnitude more energy efficiency, depending on the use case. Okay, so if you want to know more about this, you can read more about it. My intention is not to give you the exact details over here, but to show you the potential benefits that you can get. So today we're losing a lot because we're not doing this, if you will. Okay, so that's not enough. Unfortunately, this is a very fast-moving field. One of my other students, a PhD student right now, Joel, he figured out that this genetic algorithm has a lot of inefficiency. So actually improved upon it. And he basically provided a much better implementation. So you can actually improve on top of that by 6x, for example. So the numbers you can get by understanding the algorithm and co-designing it with hardware is actually very interesting. And this is actually implemented on CPUs and GPUs, and it's still better than GenAs. Okay, a lot of these are open source. If you're interested in working on this, you can find this. This is the latest work that I know of. It was published in March 2023, actually. You can find it online, you can use it, and very open to feedback, actually. We received a lot of feedback even before publishing it by putting the source code online. Okay, I will skip some of this. And you can also find talk videos of Joel talking about it. Now, let me give you some other applications. So this problem is becoming harder, actually. This is the easy part. What I just told you about is this computation, that's easy. Right now, what's happening in reference genomes is they're becoming more graphs. Why? Because people are saying, we don't want a single reference genome representing the entire human species. It doesn't make sense. The entire human species is there's a lot of variation, even though it's small, like 9.9% difference. Still, we want to represent differences so that we don't want to have biases in the reference genome. There's significant difference, for example, between the genome from Africa versus Europe versus America, etc. So we want to represent the human genome as a graph. Now, this actually makes this a much tougher problem because genome is not a linear sequence anymore, linear sequence of text. It's really a graph. Basically, you connect these bases with hops to each other, and it could actually be a very complicated graph. So this is what I basically pictorially it looks like this. We were looking at linear sequence matching before. Now we want to actually match a read, which is linear still, to a graph. And this is expensive, much more expensive. We have graph alignment tools today, that's software, but they're also slow. So basically, this is called the sequence to graph mapping problem. Essentially, we wanted to design better algorithms and accelerators for it, and we're working on both of them. And this CGRAM is the first accelerator that works for both sequence to sequence matching as well as sequence to graph matching. And again, I don't have time to go through the details. It's a fascinating field over here. This is a more complicated version of the sequence to graph mapping pipeline. You need to construct the graph, but these are offline pre-processing. And once you construct the graph, you index it. And then online, when you actually get the reads, you need to find where this is matched in the index. And then you do a lot of filtering, chaining, clustering. If you don't know about this, don't worry, you can read the papers and figure that out. That's why we write actually these overview papers. And then eventually you need to do the sequence to graph alignment. So there's a long pipeline associated with it. So we designed hardware for this. Again, I don't have time to go through all of the components over it, but essentially we have an algorithm, we accelerate the algorithms using specialized hardware. And in the end, you get significant benefits. Sorry, I'm going through this maybe relatively quickly, but I don't want to cover a single algorithm. The other reason I'm doing this is because algorithms are changing and the hardware is changing. It's important to really be on top of the field so that you can actually improve algorithms and hardware at the same time. So this is, to our knowledge, this is the state of the art in terms of sequence to graph mapping, unless there's a paper coming up in the next few months that I don't know of, which may be happening, of course. But you can see that the speedups are huge compared to software. VG, for example, or Graph Aligner, these are the state of the art software tools. They have different capabilities, so you need to compare to both of them. But you can see that the speed up and the power consumption is much lower compared to these software tools. And there are very few hardware tools over here. This was the first hardware tool that accelerated sequence to graph mapping. If you're interested, you can watch download video and some of the other works that we're doing. Okay, so let me give you some more problems, if you want. So far, I assumed text, right? I said reference genome is text. But who said that, right? Whenever you actually sequence a genome, what you get is electrical signals. This is a nanopore machine that you have over here. A nanopore machine is essentially a nanoscale hole. So if you look at this nanoscale hole, it's made of graphene actually. You have the DNA strand, DNA passes through this nanoscale hole. And while the DNA is passing through this nanoscale hole, this machine is measuring currents. Essentially, what you have is electrical signals. So why do we even convert it in the first place to ACGPs? Why don't we directly analyze these electrical signals? This is something that we're working on right now, but this changes the problem almost completely. That's why I didn't want to spend a lot of time on all of these algorithms, but because I believe the future will be very different. Maybe we're going to actually do this analysis on raw signals. This is called raw signal analysis. I'm going to mention a paper that's coming up in July in the ISMB conference. Okay, so basically we have mechanisms to accelerate these base calling. So there's a lot of machine learning involved in converting these signals to ACTGs right now, but we want to get rid of that inefficiency of converting these signals. Why don't we base all of our analysis to these raw signals as opposed to ACGPs completely? Okay, how much time do we have? I feel like I've been talking a lot. I want to have some time for questions also, so. Okay. So, okay, maybe I'll give you very quickly processing in memory and then I'll conclude. Does that sound good? So I want to give you processing in memory because this is something that's also happening. Today, we're actually processing things, moving things into a compute chip and accelerating them. But is that the right thing to do? We're actually generating a lot of data and storing them in storage DRAM. Can we actually process them where they sit? So that's the idea of processing in memory. Instead of processing things in the processor, CPU, GPU, FPGA, whatever your favorite processor is, heterogeneous machine learning accelerator, keep the data inside the memory or storage and ask memory, can you actually do the sequence matching for me, for example? And the memory, without moving the data, does that sequence matching and gives you the results. This is the general processing in memory. So there are many questions over here, which I don't have time to talk about. This basically changes the computation model significantly. And it has its own challenges, of course, but it's a great fit for genome analysis, actually. Essentially, we need to design mapping and filtering algorithms that fit processing in memory. And that's what we've been doing. Let me give you an example of this. So the sneaky snake algorithm that I mentioned earlier. it tackles the read mapping, right? And it is very much memory bound in the end. It has to move a lot of data from main memory back and forth to the CPU. So our solution is to perform read mapping near where the data resides using specialized logic. So we carefully redesigned the accelerator logic of Sneaky Snake to exploit near memory computation capability on real FPGA boards that use high bandwidth memory. I'm going to show you a picture of this now. And once you do that, you get even higher energy efficiency and performance improvement on top of Sneaky Snake, for which I showed you a whole lot of energy and performance improvements right earlier. So this is basically the system. This is a general purpose CPU. This is an FPGA board. And you can offload computation to this FPGA board, which has very high bandwidth access. Essentially, this is near memory logic. And you get significant performance energy improvements. Again, there are a lot of details over here on how you do that, which I will leave you to the papers. So that's the paper that explains it. I'm not, and there are other emerging technologies like 3D stacking that look like this, where you can actually do the processing underneath memory over here. We have works on that, which I'm not going to talk about. I will mention this work, and then I'm going to go to the conclusion slides. I think this work this is very important because going forward, we need to push the processing to the sensor side as much as possible. If you cannot do it in the sensor side for various reasons, because eventually you need to store the data, you need to reuse the data, etc., you need to push the processing to the storage and memory side, because that's where the data resides. Right? Moving the data every time to the CPU, to the processor, it doesn't make sense from an energy efficiency and performance perspective. So in this work, we want to tackle this. So if you're doing metagenomics, for example, you have really terabytes and terabytes and terabytes of data, petabytes of data in some cases, depending on how much data you have. It doesn't even make sense to bring that data to main memory, in my opinion. So in this work, we designed a processor in-storage processing system. I'll go through this quickly because I've already mentioned all this. Basically, you have a huge data movement overhead from storage. This data movement overhead remains even if you do in-memory processing. So if we have a lot of improvements over here, accelerators, filters, etc., that I talked about, those fix the computation overhead. But the data movement overhead relatively becomes even larger because computation reduces, data movement overhead still remains. So our idea in this work was to do filtering in the storage system, similar to the filtering that I discussed, as opposed to moving all of these data that you need to process, filter them with some intelligent filters that are hardware accelerated over here, so that computation units gets only the very small fraction that it really, really needs to get. So the question of course becomes, what can you filter? What are the algorithms? Initially, we looked at exactly matching reads and obviously not matching reads. These are easier to detect. And once you do that, again, I don't have time to go over this. You can read it when you have time, or if you're interested. Basically, the difficulty is catering to the different behavior of the workloads, as well as limited hardware resources that you have in the storage system. But let me give you the key results very quickly. This reduces the data movement overhead significantly. And again, we see significant speed ups compared to the best prior mechanisms, up to 33x, for example, in terms of performance, and up to 29x in terms of energy reduction. So once you go into the storage side, I think there's a whole world that can improve system performance even more. And I think I'm a strong believer on the storage-centric computing for these huge applications these days, because based on all the data that we have seen, moving the data out of the storage and SSD to a main memory is extremely energy costly today. We don't even want to go to main memory, but we don't want to go to storage at all. Or we don't want to move data from storage at all. Okay, so if you're interested, that's the paper. There are a lot of interesting other papers in processing in memory that I'm not going to talk about. You can find out more about them. And also we have tutorials, but you can find them. Let me jump into the, I'm going to skip all of these. If you're interested, you can take a look at them. Oh, sorry. Let me conclude, and then we can take some questions, because it'd be great to have some questions. Okay. Yeah, I'm going to skip these. These are new technologies. I've already talked about some of these. Raw signal analysis, for example. But maybe I should mention this. If you're interested in raw signal analysis, I think this is a very fascinating direction. Okay, that's much better that way. That's a very fascinating direction. I think this is really the next frontier, in my opinion, in sequence analysis. And we have this paper coming up in the major molecular biology conference, ISMB, that talks about how to process large genomes in real time, just by looking at raw signals, not even converting them to AC. I think this is going to be very, very interesting. And I think this is also a great fit for storage-centric computation. Okay. So let me conclude by showing what is happening in industry. Industry is actually picking up some of these ideas. This Illumina FPGA board, Dragon. They have it. They don't talk a whole lot about it, unfortunately, but they do some variant calling and acceleration pre-alignment filtering, like we've been discussing. which is interesting. But they don't talk a lot about it, but I believe they have some of this programmable if you buy their machines, of course, and work with them. But these are expensive. You're not going to pay only $1,000 for this. Do you also have Illumina machines here?
It's not Illumina.
Okay, I see. But did you use the Dragon platform, FPGAs, or no? Okay. Yeah, sure. If you have the time and patience, this may be very interesting, certainly. And it's not just Illumina, NVIDIA, which is known for GPUs. They actually acquired a startup that does GPU-based acceleration, which is, I think, shorter term in my opinion, but it's still interesting. And NVIDIA has been adding these dynamic programming instructions to accelerate edit-distance computation, for example, and other things. And then there's the BioNano and NVIDIA collaboration, which I'm not going to talk about. Okay, let me conclude by recalling our dream. This was our dream. We wanted an embedded device that can perform comprehensive genome analysis in real time within a minute. We still have a long ways to go. We actually came a long way, but we still have a long ways to go because we need to tackle even more problems in terms of energy efficiency, performance, and we are not even talking about security and privacy yet. And we have a huge memory bottleneck to tackle, and that's what we've been tackling. So I think I've already said a lot of this. Hopefully I've convinced you that it's an important and interesting problem. We covered various recent ideas from my personal journey, especially. I believe there are many future opportunities that exist, especially with new sequencing technologies that are going to evolve over time. Even nanopore is evolving over time, and especially with new applications and use cases. And I believe raw signal analysis is one case over here, and also reference genomes becoming much more dynamic. So if you look back 5 years ago, reference genomes were very static. Right now, they're becoming dynamic. You actually have graphs, and they're also growing. People want to update reference genomes quickly. I think those are actually going to make this even more challenging into the future. So there's a huge bright future over here. I will conclude with one thing over here that I want to get to, but this is not getting to. Yeah, there's a lot of information out there, but I'll acknowledge my group more than anyone else because they're the ones who did the hard work. I, of course, worked with them in some cases very painfully, but in the end, the painful work leads to a lot of interesting ideas, in my opinion. And these are some pictures from them. You can find more pictures. We're going to release a 2023 edition of our newsletter in 2 days, probably, if you look for our webpage. Okay, you can find almost all of this online. A lot of these are open source. People are using them. People are updating them. We're very open to getting feedback on these. And this is where I'll end. Thank you for your.
Professor Mutlu, first of all, I would like to thank you for this very inspirational presentation. I'm still processing what you have said. But yeah, so let me just briefly introduce myself. My name is Onur, and I'm a full-time engaged academic researcher here at the Center of Excellence Hub. And also I'm a PhD student at the Technical University of Vienna. My research is partly related to the bioinformatics and biostatistics. And I'd like to hear from you, what is your opinion on how does it compare to the opportunities we will have in the perspective of remapping is connected with the large language models?
So that's a great question. This is something that needs to be explored more. I believe there's a lot of opportunity here, and I think multiple people also think that way. I think going forward, we need to think about how to take advantage of those large language models in the bioinformatics domain, because there's a lot of advances clearly. And I actually believe that there's a huge opportunity. So we need to actually be working on it. I cannot say more because we're actually also working on it, but we don't have results at this point. The worry I have over there is how to do it efficiently. Because clearly large language models we have today are powerful, but they're not efficient. They're actually exactly the opposite of what I would call efficient, right? So can we do it efficiently is the interesting part over there. Can we somehow specialize the bioinformatics domain and also figure out how to do it with more efficiency? So that also will require hardware acceleration, in my opinion, algorithm hardware for design. Thank you.
Well, from my side, my name is Onur, and I come from faculty for international economics, finance, and business here at the University of Memory-Centric. So this is not exactly my scope. It was very tempting for me to follow everything that you were saying. But my question will come regarding your university biography. So what I want to ask is when it comes to everything that is the field of the research, like what made more impact on everything you have achieved? Like your research work in America or here in Europe, especially in Syria and the places where that would be.
Okay, so let me try to understand it. You're asking what research I did had more impact on the world, or you're asking
like what
on me? Oh, okay, I see. Basically, I would say everything is a collective experience. So certainly, and the research I did at different places didn't happen at the concurrently, right? It happened at different stages of life, different times. So I learned from everything that I've experienced, and certainly initial formation, the years where you study the fundamentals at a university, for example, and figure out what is interesting to you. Those formative years are extremely important. For example, when I went to University of Michigan for my undergraduate degree, I went there to become a computer engineer. But then I took a class, Psychology 101, and I said, okay, I'm going to do a degree in psychology also, because I love the teacher who taught me psychology. I later took another class from him, actually. He was a very, let's say, brilliant teacher, I would say. But I did my PhD with another teacher whom I took my first computer engineering class from, was Yale Pass. So clearly those initial years had a lot of impact. But it's very hard to distinguish, okay, what had more impact, less impact, right? I don't know if that answers your question,
Yeah,
but
difference, not
yeah. Yeah, certainly. I mean, there are differences in different places, right? I did my PhD at Texas, I did my undergraduate degree at Michigan, and then I was at Microsoft Research for several years, starting the group, and then Carnegie Mellon for several years, and then ETH for several years right now. They're all different places, but you can do great work everywhere. Sure. Yes.
So, let's say exact questions that I'm interested in. First of all, we are using genome sequencing for analysis of bacterial species. So we have discovered some new species from a neighbor thanks to this technology. However, I'm interested in data processing for the sequence alignment. We are mostly using pipelines and softwares or the platforms that are open, online, and very slow. However, I know that there are some platforms available for purchase, and we are now thinking of purchasing one software for the sequence processing. Data processing, so maybe you have some kind of recommendation. But in the light of your latest talk about Core and turning the whole reading into the electrical signal reading, how you will make the reference, or how do you transform now all the mass of the data that we already have into the reference electrical signals? So, of course, I'm not guaranteed, so maybe this is easy because I'm just interested.
Yeah, sure. I mean, that's a great question, basically. That's why it's very disruptive, I think, because everybody is doing text-based, and we have developed a huge database of text-based references, right, in the world. Raw signal will change all of that, and it will clearly take some time for us to change everything to raw signals. But there are some analyses that become much more efficient because of that raw signal. And we are still discovering the algorithms also, like this paper that I mentioned that's going to appear in July 2023 is one of the earliest papers that look at this sort of algorithms. So we're still at this infancy. I wouldn't recommend going that direction immediately if you're using these algorithms, but over time, in my opinion, we need to get to it, especially in those cases where text-based search is not efficient. I have to talk about it further. There are a lot of research over here that could be done also. In terms of your first question, whether some specific software package is better or not, I don't want to take sides right now, but I'm happy to discuss it further. Say it again. Yeah, it will stay between us. Yeah, we can talk between us. Yeah, but certainly there's a lot of optimizations that you can do, right? I assume you're using a lot of open source tools right now. a lot of them are not very optimized. In fact, that's one of the reasons why I went to this area. So one of my friends at University of Washington Genome Sciences Department was developing these tools, and he knew that I was interested in accelerating everything, let's say. So he came to me and said, can we actually do this tool and accelerate it? And that was our first paper, Nature Genetics. What I did was very simple in the end, very simple acceleration. So actually, if you know what you're doing, you can get orders of magnitude speed up in even the software. So my guess is the tools you're examining, if they're used by people, they're probably getting orders of magnitude speed up. That's probably a worth investment, but of course, investment decisions are different from scientific decisions in the end, right? Sure, you're welcome. Please go ahead.
My name is Onur. I'm a professor here. Texas A&M University, so very competitive.
Okay, nice. Not with me.
Not with me either. Yeah, I didn't hear much about it. I was wearing the cloak and the shirt with the colors of the Texas, and I learned my lesson.
Yeah.
But the generic question that might be interesting for the group here. What is your take on open access publishing and open source software? So what are the benefits of these advantages? So that's one question. And another question I don't remember is your,
Okay, sure. So open source software. I'm a big believer in open source. That's why we have all these tools as much as possible open source. I think this is really important for everything, let's say, scientific community, worldwide development. I don't think it makes sense to hide things. I clearly understand that maybe you want to open source a little bit later so that you can actually develop ideas because you've made a lot of investment, for example, to build an infrastructure. So I'm a big believer in open source. There's a lot of benefit. Open access, I think everything should be open access. I don't like the fact that publishers are charging fees for it. That's the huge downside of open access, I think. That goes against open source, right? I mean, nobody should charge anything open source, but open access, I think publishing fees are the let's say, top part, let's say, and some of the fees are huge. I don't know if you were referring to that, but
no, I'm all for open access and sort of, but sometimes I'm having struggling here to with students, because they not paying the software, they are not open for switching to the open source something. But just the regional problem.
I see. Okay. Yeah.
My other question was also more personal question than technical. How do when you are hiking in the mountains with your friends?
Yeah.
How did you come to the question we want to do a genome sequencing under 1 minute? That was kind of.
Well, that's the mountains, I guess, are the inspirational parts.
I don't understand the other community goal.
Yeah.
Done in embedded,
Yeah. So basically, we were developing this mapper, read mapper together. It was purely software, and we were having a lot of performance issues, etc. So we were brainstorming on how to potentially.
So all the group was from the same.
Well, it was mainly two people, actually, me and my friend, but essentially it was a good brainstorming session based on that. But yeah, we didn't solve the particular problem that we were brainstorming about at the spot, but we had a much more longer term direction that we thought would be very interesting, which essentially seeded a lot of the research that I talked about.
One more question.
One is more serious and the other one is not very much. The first one, when you see a mismatch on a single character level, how you know it is from a mutation or from the fact that the chemical process was not ideal.
Okay. Maybe I can repeat it very quickly. Basically, when you see a mismatch at a single character level, single base, how do you know that it's because of a mutation? It's real versus it's not a machine error, right? Because of some chemical process of the machine or electrical signal misdetection, etc. So that's a great question. Something that I did not tell in all of these is you need to build confidence. So how you build confidence is you actually don't sequence a genome only once. You sequence it many times. This is called coverage. in bioinformatics terminology. And in some cases, if you want to get very high accuracy, you need to sequence the genome 30 times, for example, 30x coverage. So a lot of these algorithms have another step afterwards if you want to get high accuracy to avoid those errors, where you build confidence by trying to essentially see whether that is a real mutation or that's because of an error. So hopefully, if it's because of an error, one time you sequence, you get an error in this one, but the remaining 29 times you sequence, you don't get an error in that character. You may get an error in some other character, but hopefully you don't get that error in that character in the other 29 times you sequence. Yeah.
And then the other one about onion. Well, you said humans 3.2 billion, onion 60.
Yeah, exactly. If you go back to the exact number, I don't remember, but yes.
So how would you explain the difference? Or maybe this is a sign that one day onion will become a lot more sophisticated than human.
So this is not a serious question. I don't need to answer it perfectly. Basically, I don't think we know exactly like what's that. So maybe the length of a genome doesn't necessarily have a perfect relationship in terms of the evolution of species so far. I don't think this question is answered at this point. But again, I don't. It's not my expertise also. Probably you should ask this question to some biologist.
Looks like onion has a lot more potential for something very, very sophisticated than human.
That is true. Actually, yes, in the end, that is true in terms of the encodings you can have. Clearly, it's much larger. But the interesting part in, this is the part that I cannot comfortably say things about because my training is not that, but there are parts of the genome that are coding and non-coding, meaning some parts are essentially what we believe is useless. We're not sure, of course, absolutely, because we don't know enough. But even human genome has a lot of parts that are non-coding. Maybe onion has even more parts that are non-coding. I will study that, I don't know. So yeah, I don't know, basically. The garbage one. yeah, exactly. Maybe evolution caused some of that to be completely garbage and it cannot be used anymore. I don't know. Yes, please. Oh, there are more.
Thank you for a very interesting speech from bioinformatics area. I'm Luka Filipovic, working at University projects regarding XTC. So IT guy, which is working with the full stuff. For me, it was more biological story, and it was interesting how to motivate you to work in biology area. My question is the CPU strong enough for shorter genome sequences, or we must use the GPU or EG or similar?
Yeah, so that's a good question. Basically, whether you have short versus long, in the end, it doesn't matter. The CPU is not good enough. If you have shorter sequences that is generated by these sequencing machines, shorter versus longer, both of them are unfortunately too much for a CPU. It's expensive for different reasons. For shorter sequences, actually, there's a lot more information that you need to do because we need to match a large genome. And again, those are costly. If you specialize, I'm using an FPGA, or if you parallelize, I'm using a GPU gain. For longer sequences, there are a lot of errors, so the edit distances need to be higher. So even if your sequence is longer, and maybe you think the CPU is okay, but usually not. So in the end, unfortunately, CPUs are not the best. Yeah, oh, yes, let's say.
Thank you very much.
Sure, you're welcome.
Hello, nice having you. Hello, it's nice having you here. It's nice having you here. I just wanted to ask something related to the maybe only a question.
Okay.
So if you know that, I'm not coming from this field, so if you know that the genes are constantly changing, so they are switches which turn on and off constantly in time, in matters of seconds, minutes, or hours or days, you are actually analyzing the picture, which is frozen in time, just a frame, right? So how can you know that the results which you get are actually meaningful? Because I don't know if you understood. The genes are constantly changing, like
What do you mean by changing?
they are constantly turning on and off.
Uh,
That's what I know. I'm not coming from this.
Well, they're not changing in the sense that physically your sequence is unique. Basically, it doesn't change. So the DNA in you is not changing. There may be mutations, of course, but it's not that frequent, right, in your body. It's a unique sequence that you have, which is different from the unique sequence that I have, which is different from the unique sequence that Velcro has, which is different from the unique sequence that Naim has. So I don't think they're constantly changing. What may be changing is like a virus, like take COVID-19, right? As it mutates, and it may mutate very quickly, of course, right? It's caused these different variants, right? And that was the difficulty with COVID. Basically, it mutated very, very quickly. So you had so many variants around. So I think what you're referring to is real.
So the genome is always
For a given individual, the genome is the same. Mutation may happen, but they're very, very infrequent. Like you get exposed to some radiation and you get mutations in that case, right? But for a given species, yes, mutations happen, especially in viruses that are quickly adapting to the environment. So I think your question is applicable to that species level. And that makes the analysis difficult, of course, right? That's why we've had the difficulty of this this COVID RNA is constantly mutating. How do you develop, let's say, good or good enough vaccines for it? Or how do you even identify what variant you're dealing with if you don't have a vaccine for it? Yeah, so basically the difficulty that you mentioned is real, but it happens at a more species level. Yeah. Now it's time for the exam then. Oh, another question about echo. If somebody wants to delay the exam.
My name is Maina March, and I have a PhD student that is in international economics, but also the grant holder from the PhD studies from the Ministry of Science. I just one technical question, but because we have a problem here to become national congress and we and also the center of excellence in microbiome for this to get education student around this center of excellence in this national congress. So I have a question, can you get the presentation?
Yeah, absolutely. Who do I send it to? I can send it to Velcro or. Okay, do I have her email? Okay. Oh, okay. This is my own laptop, so I need to send it. So I'll leave the computer, okay. Then I won't be able to give the presentation tomorrow, but I'm happy to send it, yes, as long as.
Thank you for your presentation. It's great honor.
Thank you. Thank you.